When RNA polymerase type 2 first transcribes mRNA, it doesn't look like this nice colorful one here. It's first called heterogeneous nuclear RNA, or hnRNA, and needs to go through a few processing steps in the nucleus before it's officially an mRNA. So first, a 7-methylguanosine cap is added to the 5' end, which is literally just a guanosine molecule with an extra methyl group on it, but it's connected backwards, so its 5' end is connected to the 5' end of the mRNA by 3 phosphates. Next, the sequence AAU-AAA at the end of the 3' UTR is used as a signal to add about 200 more A's to the end, which is called polyadenylation, and forms the poly-A tail. Both the cap and the poly-A tail regulate nuclear export, prevent degradation by exonucleases, and promote translation. The third thing that happens is introns have to be spliced out, so you're left with just the normal coding sequence. So how is this going to show up in your exam? Well, they might give you what I call the laboratory vignette, which is something like, you're working in a lab and you isolate an mRNA molecule from some cells, but you notice that for some reason it has no 5' cap on it. What does that tell you about this mRNA? And they'll give you some choices that would seem plausible to some med students, but not you, of course, because you've seen this lecture, like maybe an early stop codon, or a mutation in the promoter, or maybe a mutation in the start codon. But you know these aren't going to affect your 5' cap. But since the 5' cap is only added in the nucleus, genes that are encoded in the mitochondrial genome would never be capped. So there's your answer. Alright, flash quiz time. What are the 3 modifications that need to be made to the hnRNA before it can be exported from the nucleus? So those would be adding the poly-A tail, adding the 5' cap, and splicing out the introns.